it's fabulous to hear these talks and stitch these pieces together. So, and actually, sort of between Julie's and Howard's talks, you are now set up perfectly to hear another facet of the XI. And so I'm going to suggest that XI does more than cause autoimmune disease. I'm not going to dispute Howard's claim, but actually we'll get in. Maybe we'll, I won't. I think it's a fabulous piece of work that Howard has just presented, and it was really interesting to hear from Julie about sex-biased gene expression on the X chromosome. So let me introduce you to my lab is very much on a mission. And it's one that we've sort of come to focus on over the last 6, 8, 10 years, and that is to discover the molecular bridges that connect the sex chromosomes to sex differences in health and disease. Now, I would say that Howard just presented a very direct connection, which is, take a sex chromosome and demonstrate it to be an autoantigen. I can think of no more direct way. I'm going to talk about somewhat less direct ways that might require transcription and translation and things of that sort. But I will assert that all biologically based differences between males and females in health and disease, those that are not driven by environmental or social cues, for example, I will assert that all biologically based sex differences between males and females trace their origins to the sex chromosomes. I'll let you think about that and think about whether you agree with that. And let me point out here that even this, where do the sex hormones come from? They come from the gonads, whose fate is determined by the presence or absence of the SRY gene in the Y chromosome. So I am not, by saying that all things trace their origins as sex chromosomes, I am not excluding hormones. I am including them, but just reminding you that in the larger scheme of things, including development, the sex chromosomes are calling the shots there. But I will assert that with some limited exceptions, including the case that Howard has just presented to us for autoimmune disease, and with the exception of things like X-linked recessive disorders, we generally do not know how to think about the connection between the sex chromosomes and sex differences in health and disease. So it's kind of, it needs to be there. We need to build those bridges. So in that context, I want to invite you to join me in rethinking the inactive X chromosome and think about the molecular origins of sex differences in human biology. And so the heroine of my comments will actually be the XI. And I'm just sort of thinking about playing off, right? So we have the devil as XI in autoimmunity, and I'm going to think about, okay, let's go there. Let's see what XI is about. And actually, it was funny. I first heard Howard talk about this beautiful story at a meeting we were at in Berlin a year and a half ago. And I was one of the early speakers, and it was a meeting on X chromosome and activation. And I remember getting up in front of the audience and explaining, most of the folks assembled there are students of the mechanism of X chromosome and activation in mice. And I said, I am so glad that you're all working so hard on explaining the mechanism of X chromosome and activation. I want to tell you that in my lab, we do not study the mechanism, we study the product of X chromosome and activation. And people looked at me like, why would you do that? Why would anybody study an inactive chromosome? Anyway, so let's see what happens here. So I'm going to go back to some things that Howard introduced us to. So here is a 1949 paper by Barr and Bertram. We talked about the Barr body. There's a real person named Barr, right? And who actually worked at the University of Western Ontario in London, and where the Barr body was discovered. And it started out as just this little speck I like this, in the neurons of female but not male cats. That little thing that the arrow is pointing at. In any case, so this was the first, and in this paper, they had no idea that this was an X chromosome, let alone an inactivated or heterochromatinized. So, so that's 1949. I'm going to make the point, this is a field with an old intellectual history, which makes it both wonderful and fraught. Yeah, so hence the term Barr body synonymous with XI. Or now we could say autoimmune autoantigen, right? So, and then along comes Mary Lyon in 1961 with this beautiful paper. If you haven't read it, I really recommend it to you. It is all of 1 page long, okay? And it has no primary data. It does cite the tortoiseshell cat, but basically the paper, so what she says is that the evidence of mouse genetics indicates that the Barr body, my insertion, can be either paternal or maternal in origin in different cells of the same animal. It can be a mosaic, and that it is genetically inactivated. That phrase, she would actually within a year say that she regretted. Because hence the term inactive X chromosome, XI, and X chromosome inactivation. So within a year, she began to realize that there were some problems in extending this idea to humans. And she mused a little bit on whether she should have had a different choice of words. But I would say we're still laboring under this now deeply entrenched term. So, okay, so now I introduced you already to the human X and Y chromosomes. I'm going to give you another layer to that introduction. So in conventional genetic parlance, we would say that humans have sex chromosomes of 2 kinds. Now, by now you all know, what's the little green bit up there? The pseudoautosomal region, right, which is identical in sequence between the X and Y. And then we have, and we have the non-pseudoautosomal parts of the X and of the Y. And I told you that we have these NPX, NPY gene pairs. This is like I'm teaching a class and I actually get to this is like the second lecture after, right, okay. So we got these 17 NPX, NPY gene pairs. So that's what we say in conventional genetic parlance. But epigenetically speaking, and this is where it gets amazing, epigenetically speaking, human somatic cells have sex chromosomes of 3 kinds. XA, XI, and Y. The so-called active, inactive. I'm going to stop using those terms and just talk about XA and XI. And the amazing thing is that these chromosomes can exist in various combinations within the same cells. So, quick test. What is the name of, what name is given to individuals, humans, who carry one of each of these 3 in their sex chromosomes? This is called Klinefelter syndrome, right? Okay. So, very good. And actually, then that gets back to if you want to have a really cute problem set in your genetics course, you say, okay, so calico cats are always female. How do you explain a calico male cat? Right there, one of each of those 3. So a Klinefelter's cat can be a calico, right? So, Mike, I'm looking for, you collect calico cats, right? You said you have only... I don't have any Klinefelters. You don't have any calico males. Okay, so I want to come, next time I come back, I want to hear that some of you have calico male cats as pets. Right, it's right. It's very special. It's very special. So if we have these sex chromosomes of 3 kinds, what it means is that euploid human somatic cells that is sort of chromosomally normal, human somatic cells typically come in these two varieties, an XA and an XY, an XA and an XI in females, or an XA and a Y in males. Now, over the last several years, my lab has been doing deep dives into the epigenetics of XA and XI. We've heard some about that already. I'll tell you about really, we're sort of obsessed with this question. We have looked, but can find no differences epigenetically between the XAs in female and male cells. And while I'll say that people in the field would say, oh yeah, males have an XA, females have an XA, when we began to assert that the XAs were the same in males and females, we got enormous pushback because people had this sense that it should be a little bit more like flies and worms, that there should be something inherently feminizing about an X chromosome. But really, it appears to be, XAs appear to be epigenetically uniform. XA is not, there is nothing female about XA. So the chromosomes that differ between the sexes then in humans are XI versus Y. So I invite you so what it means is that the other 45 chromosomes, including XA, are equivalent genetically and epigenetically in female and male somatic cells. So in our lab, we've begun to refer to the XA as the 45th autosome. Not in the sense of its mode of inheritance, but with respect to its function in the mortal cells of the body called the soma. And what this means is I invite you to entertain the idea that rather than, we've said for a century that males are XY and females are XX. I invite you to entertain saying that instead of saying that females are XX and males are XY, say that females are XI and males are Y. And you might say, well, isn't that just a semantic game? I say, no, it is not a semantic game because it actually focuses the question very intently on the biology of XI and the biology of Y. So the opportunity then to discover the molecular bridges that connect the sex chromosomes to sex differences, we are, in our lab, we're translating this to the opportunity to connect XI versus Y to sex differences. And the first task then is to actually characterize XI, the product of X chromosome inactivation, which what I'll try to give you a sense of is my own experience is over the last few years, we've come to appreciate that XI is a really, really amazing chromosome in all the senses that Howard has explained, but in some other senses as well. So let me just begin to introduce you to my obsession with this version of the 46th chromosome. Oh, and Howard already mentioned the N-1 rule, something that's very powerful, which is, and this was known since 1961, that every diploid individual walking the planet has one XA, and no matter how many other X's you have, they're XIs. Now, you may say, well, why do you say no matter how many X's you have? Well, I'm going to tell you that there are people walking the planet with as many as 4 X chromosomes. It's not tolerated on any autosome. And it turns out this becomes very important in some of the experiments, in some of the analyses we've done. So I showed you this picture before to highlight the role of Britt Jepsen-Darbeloff. When this photograph was taken, I'm going to tell you a good bit about the work of Adriana San Roman, who was a postdoc in the lab, now actually runs her own lab at Duke. And I will not have time to talk about the work of a fabulous graduate student, Sahin Naqvi, who was then a postdoc here with Jonathan Pritchard and Ioana Vysotsky, and is now a faculty member at Harvard Medical School and Boston Children's. So again, the Stanford, MIT, Whitehead connections just keep reverberating. So to get to Adriana's work, so basically over the last 6-7 years, we have been having so much fun exploring Xi and Y, both in vitro in cultured cells and in vivo in cells taken directly from the body, by studying the wide range of naturally occurring sex chromosome aneuploidy in our population. And so this involves studying individuals with 1 to 4 X chromosomes and 0 to 4 Y chromosomes, and setting up experiments in LCLs. Julia just told us about studies in LCLs. This is a different dimension of that study, and then also primary fibroblasts. And I will tell you very briefly, for the sake of time, I will get to describing a little bit the studies of another fabulous postdoc, Laura Blanton, in the in vivo side of this proposition, looking at immune cell populations, sort of anticipating, Marcia, your next installment of this meeting, looking at immune cells. And so in this case, looking at, and here we show the sex chromosome constitutions of individuals with 1X. You notice they can have variable numbers of Y chromosomes. You might say, my gosh, I never imagined there'd be such variation. And then, so these are 1, 2, 3, 4 X chromosomes, which means, what's the number? If you have 1 X chromosome, you have 1 XA, 0 XIs. When you get up to 4 X chromosomes, it's 1 XA and 3 XIs. So it's actually a linear series. And it's like an allelic series for the geneticists here, right, across with respect to the number of XIs, and have a Y chromosome copy number series. And it turns out this is a setup for using very simple mathematical linear modeling to characterize in a multi-omics way. Everything we've published thus far is transcriptomics, but we've got multiple other omics in the works. To characterize the ohms of Xi and Y, that is, they're directly on those chromosomes, but also their impact on autosomes. The amazing thing is that this makes it possible to characterize those chromosomes directly and their impact without physically isolating them. And I understand I'm saying that, and you're saying, well, what does that really mean? Well, I think it actually, we sort of have to do it in the lab. You sort of have to do this to really come to understand what it means. But I'm going to show you some of the data so you get a sense of actually how relatively straightforward some of this is. Okay, in fact, I'm going to show you transcript. I'm going to show you RNA-seq data from just 3 genes on the X chromosome in the lymphoblastoid cell lines, so you can see what this looks like. So here is a gene, PRPS2. This is a gene that is, like most genes on the X chromosome, expressed only from XA, not expressed from XI. Each dot is an unrelated individual, lymphoblastoid cell line from one unrelated individual. And you see there's some noise, but basically as you add X chromosomes, it's pretty much a horizontal line. There's no slope. Now, let's put PRPS2 aside and let's go to exist. Let's see what it looks like. Okay. Good start. 1 X chromosome, that X chromosome is an XA. Exist is not expressed. Okay. I'm not making this up. It's all really basic. I mean, exist is really not expressed in individuals with 1 X chromosome, no matter, like, you can add 4 Y chromosomes if you want to. It doesn't make any difference. But now let's see what happens as we add XIs. It goes like so. It goes up. It's not, it's not really tight, but it goes up. Oh, that's nice. So now let's go to another kind of gene. And this is KDM5C. It's that gene I showed you in the opening comment as an example of an XY survivor. So this is, now this one still takes my breath away when I look at this. These are unrelated individuals. There's no inbreeding here, right? And I say you could, you remember when you were taught in some early science course to draw a line and calculate a slope and an intercept? You could do that with this, right? And in fact, if I just told you the level of expression of this gene, I could pretty much tell you how many X chromosomes that person has with reasonable confidence. And so this is what it was this kind of image that really inspired us to think, oh my gosh, we really could use linear modeling. So note the transcriptional modularity of XA and XI. I mean, so realize the intercept is XA and the slope is XI. You're adding XIs, right? And I'll make the point that this is partly where I said an XA is an XA regardless of whether it's in a male or a female. This is intermingling of data from individuals with a Y chromosome and therefore testes and individuals without a Y chromosome and therefore ovaries. Doesn't make any difference. You get the same slope and intercept. And so for each gene on chromosome X, Adriana could then, in lymphoblastoid cell lines and also in fibroblasts, measure the slope and the intercept. And then just, the problem was these different genes were expressed at different levels. But it turns out that if you take the slope and divide it by the intercept, you come up with a dimensionless number that is the fraction by which Xi adds to XA's expression. We call this thing delta E sub X. It's turned out to be, for us, a very useful new metric. So again, it's just slope by intercept. And I'll just show you now the distribution of delta E sub X values for all of the 357 genes that were expressed in LCLs. Each of them has a characteristic delta E sub X value. Most of them, of course, are around 0 because they're not expressed from Xi. So nothing changes much for most genes. But some genes, like I had that KDM5C, right? It's got a delta E sub X, that is a slope divided by the intercept of about 0.7. And anyway, there's exist, it's over around 1. So, basically exist is not, yeah. So interesting, okay. And then, and so the delta E sub X values range widely among the chromosome X genes. And you see there are even some negative, there are even some genes that have negative. As you add X chromosomes, their transcript levels are falling. We don't have great molecular explanations for any of those as yet, but looks like pretty solid. Altogether, 38% of expressed chromosome X genes have a statistically non-zero delta E sub X value in LCLs or fibroblasts, which is about twice what the prior literature predicted. And that is in part because there are two different processes at work here. So combining the delta E sub X metric with, with allele-specific analyses, revealed two modes of chromosome X regulation in humans. So first is what we historically call X inactivation. That is transcriptional attenuation. I wish Mary were here, Mary Lyon were here, and we could have a negotiation and stop calling it X inactivation and call it maybe X attenuation.
Also A.
XA, yeah. Oh, yeah, so we'd have to have a different abbreviation. Thank you for, I knew there was a problem, right? XAT, I don't know. Should have thought of that, yeah. So, but there is this transcriptional attenuation of XI, and I'll make the point that we find virtually no genes on the X chromosome that have a delta E sub X of 1, which is to say, the second X is always expressed at the XI expression never actually meets the XA expression, which is partly why we no longer refer to escape from X inactivation, because the term escape, in my experience, conjures up in the mind of people hearing it for the first time. Oh, expressed just like it's on XA. And quantitatively, that is essentially never the case in our experience. But there's this transcriptional attenuation of XI in cis, from a geneticist's point of view, what we call X inactivation. But it turns out we also have lots of examples now of genes expressed from, proteins expressed from XI modulating the expression of genes on XA. Now, this shouldn't be terribly surprising. I mean, these are chromosomes. They're made of DNA. But I think for a long time, people thought there's something unique about gene regulation on the X chromosome. But it's not. So there are genes, there are proteins that are emanating from, ultimately from XA and XI that are working in trans. So these cis and trans effects differ widely in magnitude across the chromosome X genes. So it turns out you got to go gene by gene to actually understand this biology. All told, 38% of expressed X chromosome genes change expression levels due to one or the other or both of these processes. And since XI modulates XA genes in trans, the question naturally arises, does XI modulate autosomal gene expression, and does chromosome Y do the same? The short answer, and I'll keep it really short, is the answer is yes. So there are thousands of autosomal genes that respond significantly to chromosome X copy number. This is, again, just analyzing from a different vantage point the same RNA-seq data sets across sex chromosome aneuploidy series that I've just told you. And I'm picking out, here's a gene on chromosome 22 whose expression rises with the number of X chromosomes, a gene on chromosome 6 whose expression falls with the number of X chromosomes. And now, just to make it, this was not what we wanted to see, but, so I'm now just showing you these are all the statistically significant genes across all chromosomes. What we didn't want, this is something that we weren't anticipating, and at first we didn't want to see it. But look, the Y chromosome is eliciting similar responses for these genes. And so how can it be, how can the XI and the Y chromosomes have similar effects on autosomal transcription? And are these ones that I just showed you, are these isolated examples? Turns out, no, these are not isolated examples. Here, okay, this is the image. This is the plot that Adriana San Román and I did not want to look at for a long time. Because you could imagine, if our goal is to understand sex differences, we want XI and Y to have different effects on autosomes. But look at this. I am showing you data on all expressed genes, no filtering. The log2 fold change per added chromosome Y on the y-axis, very convenient, and on the x-axis, the log2 fold change per added X. And, I looked at that. I mean, you can see the correlation. So this, it really took us a while before we were willing to contemplate the significance of this. So what this is saying at first blush is that an XI or a Y, they're kind of the same, or they have the same kind of consequence. So that's a piece of the message I want to deliver here. We were not looking for this. We did not want to see it, but it's what the data show us. It's a lovely thing to have quantitative data in a field that has had no quantitative data of this nature before. And it gets worse. So if you look at the genes that show a statistically significant X response or a statistically significant Y response, this is the Venn diagram. And now let's look at the 442 genes in the middle that share a statistically significant response to the X and the Y, and let's see how their responses map. Oh my gosh, we got an R value of 0.95. Okay, so basically an XI is a Y is an XI with regard to auto. And we say, okay, maybe look, LCLs, you like them, but they're transformed. These are immortalized cell lines. Maybe this isn't real. Well, okay, let's look at fibroblasts. Yeah, oh my gosh. Okay, so it doesn't matter whether it's primary cells, transformed cells, you can see similar effects of Xi and Y. So what accounts for the shared autosomal response to Xi and Y? Well, the good news is you heard my first lecture. So now you're prepared. You should already have the answer in mind. Xi and Y come from what were once a pair of identical autosomes. Okay, so Adriana considered three possibilities to explain this. One was a generic response to aneuploidy. Maybe this is, maybe all aneuploidies do this. And I'm just going to say she ruled that out by studies of trisomy 21. Maybe, our Drosophila colleagues said, this could be heterochromatin sinks on Xi and Y that are, anyway, I won't go into the details, but she pretty much ruled that out. And so left with the possibility, these could be effects of specific genes that are shared by Xi and Y, either in the pseudoautosomal region, that'd be the easiest place, or maybe NPX, NPY gene pairs. So being geneticists and having some freezers full of cell lines collected over the decades from individuals with structurally aberrant X and Y chromosomes, we thought, let's deletion map. Okay, so we got some geneticists here right in the room. So consider the autosomal response to be a phenotype, a molecular phenotype, and you're going to map that response on structurally aberrant X and Y chromosomes. And this is so we deletion map chromosome X and Y drivers the autosomal response using structural anomalies of the human sex chromosomes, where you have more copies of some part of the X, but not all of the X. Anyway, long story short, the pseudoautosomal regions do not drive the autosomal response. The pseudoautosomal region is where everybody wanted the response to be. It would be most straightforward. It's identical sequence, identical genes. Instead, the ability to drive the autosomal response maps on chromosome X to the vicinity of the gene CFX. Doug, you should be shaking your head, thinking Doug's familiar with the history of the ZFX. And on the Y, so there we are. A pair of zinc finger transcription factors encoded by the X and the Y chromosomes. And CRISPR experiments reveal that, I won't go into the details, I'll just summarize. It looks like ZFX accounts for about half of the chromosome X effect on autosomal gene expression. So these findings so now we're going to say, okay, but maybe this is all a series of artifacts, right? You got one transformed cell type, one primary cell type, but these are all cells cultured under uniform conditions, so it begs the question of, do these understandings extend to human cells in vivo? So basically then, do they extend to can they extend to other cell types? To find the answers, Laura Blanton generated linear models of mRNA levels in circulating immune cells of sex chromosome aneuploidy individuals. So basically taking cells directly from the blood, no activation, no nothing, just and transcriptionally profile them. And so it looks like so. It's very much what I told you about before. Take individuals from the sex chromosome aneuploidy series. In this case, we weren't able to get the samples from the extremes. We were just studying individuals with 1, 2, or 3 sex chromosomes. Having a chromosome X dosage series, chromosome Y dosage series. And I'm not even going to show you the results. I'm just going to say it's completely consistent with what I showed you in the cultured cells. So it looks like these phenomena, these insights that I shared with you are actually generalizable here across 4 cell types, regardless of whether they're transformed, primary cultures, or taken directly from the body. So again, the same linear modeling. So to summarize where we are now, and Marcia, how am I doing time-wise? I have 3 minutes, okay. So our working emerging model coming out of this is, and this is going to summarize what I've told you so far and where I see this headed, which is a working hypothesis, which of course is wrong, but I don't yet know that it's wrong. That we think that a small minority of X-linked genes are XI and XA transcribed across many somatic cell types, maybe all somatic cell types. And this XI transcriptome, which looks to be relatively compositionally stable across the body, is enriched in global regulators and strong and very much depleted in what I would call terminal effector genes. By terminal effector genes, I mean the kinds of genes that are associated with the classical X-linked recessive mode of inheritance. Things like, think hemophilia, a clotting factor that does exactly one thing, or color blindness. Genes and proteins that have a singular task. But the genes that are expressed from XI significantly appear to be genes that tickle hundreds or thousands of autosomal genes and their level of expression, or global regulators. By contrast, the XA transcriptome, remember I said the XA is the 45th autosome? By contrast, the XA transcriptome is cell type specific, just like an autosome. It includes a mix of widely expressed and tissue-specific genes, the latter of which are generally not transcribed, or if they are transcribed, at very low levels on XI. So to extrapolate shamelessly, and I barely got into this, I apologize, but I want to give you a sense where we're going. We think that a ubiquitously expressed set of XI-expressed global regulators is nonetheless going to elicit cell type specific autosomal responses. So even if, so ZFX, let's say ZFX is being sent out from XI, XA and XI in virtually all cell types, but then we get back to chromatin accessibility, which will differ in a cell type specific fashion. And so the impact of ZFX expression across the body will be a cell type specific enterprise, even if ZFX expression is quite consistent. And so a task for the future is really to disentangle the sex shared and the sex differentiating impacts of the XI and the Y expressed genes. So I'm going to assert that I believe that there are sex differentiating effects, but those are lost in the much larger signal that is the sex shared. And I'll just acknowledge here both current members of the lab and alumni who are involved in the sex differences work broadly, and the individuals whose names are boxed are the ones who contributed to the work that I shared with you directly today. And this has also involved a tremendous network of collaborators. So we're looking, I again put in the box the individuals who helped us gather up all these samples from individuals with sex chromosome aneuploidy. And I would like to thank all those individuals who agreed to participate in these studies. And finally, funding sources again, as outlined here. And I'll just name a few things I didn't have time to talk about, but if you're interested, I've got the QR code up there. If you want to grab other things that we have published or have bioarchived recently, actually just have this week's cover of Genome Research on some interesting things with one XY pair, DDX3X, DDX3Y. And we have an upcoming paper, actually our first paper in a cardiology journal. Mike, back to our, so we got a paper coming out in Circulation in the Valentine's Day Women's Heart Health special issue of Circulation. And some things then on glioblastoma and some paper from our colleagues at the Trans Clinic at Boston Children's. And I'll stop there.